Greetings to everyone. Welcome back once again to this educational channel called Bio with Janet. Today we'll continue discussing chapter 5 and we are going on to subtopic 5.2 on enzymes. So enzymes are a fascinating group of chemical substances in our human body which help to speed up the rate of chemical reactions so that life can be sustained, and it helps us to carry out our living processes very efficiently and help us to be able to enjoy life too. Okay, so they're involved in the process of digestion, respiration, and other biochemical reactions. Let's dive into the topic straight away. The learning standards for today's lesson are as follows. 5.2 Enzymes. Number 1. After this lesson, you should be able to define enzymes. Number 2. Reason out the necessity of enzymes in metabolism. Number 3. Describe the naming of enzymes with the addition of ASE to their substrates. Number 4. Characterize the general properties of enzymes. Number 5. Communicate about the involvement of specific organelles in the production of intracellular enzymes and extracellular enzymes. Number 6. Explain the mechanism of enzyme action using the lock and key hypothesis. Let's have a look at the overview of this chapter, Chapter 5, Metabolism and Enzymes. So in the last video, we have already discussed what metabolism is. That is, it comprises anabolism and catabolism. Now, this is the second video for this chapter, and we're going to study enzymes, which is subtopic 5.2. Okay, and we'll be studying the definition and the necessity of using enzymes in the body, the naming of enzymes, the mechanism of enzyme action based on the lock and key hypothesis, the characteristics of enzymes, types of enzymes which are intracellular enzymes and extracellular enzymes. And in the last video, we will study the factors that affect enzyme action, such as temperature, pH, substrate concentration, and enzyme concentration. Okay, so the third video will involve this subtopic or this content here, plus the application of enzymes in daily life. Right? So let's concentrate on the first part about enzymes. So we'll start with the definition. Right, so 5.2 enzymes. Firstly, let us define what enzymes are. This is learning standard number 1. D for definition. An enzyme is an organic catalyst that is usually made up of proteins and is produced by living organisms to speed up the rate of biochemical reactions in a cell. Okay, so there are 4 parts here to the definition, and you have to memorize it word for word if it is a definition. So first we have to understand what it means. So organic catalyst. So a catalyst is a substance that can speed up the rate of a chemical reaction. All right, but when you say organic, it means that it's produced by living organisms. It contains carbon and usually is a complex compound. In this case, most enzymes are made up of proteins, right, produced by the cell. And they are produced by living organisms to speed up the rate of biochemical reactions in a cell. All right, so they are very important for speeding up biochemical reactions so that life can be sustained. All right, so number 2, learning standard number 2, explain the necessity of enzymes in metabolism or in biochemical reactions. So in a cell, biochemical reactions have to occur at a high rate to protect living processes and to support life. For example, in order for respiration to occur quickly and to produce energy for cellular processes, we need enzymes, which are the respiratory enzymes. Okay, so as an example, respiration, which involves a series of reactions, have to occur at a high rate, okay, to produce the energy that we need for muscle contraction, for active transport, cell division, and so forth. So enzymes help to speed up the biochemical reactions so that processes such as respiration and digestion can be performed quickly for the organism's survival. And without enzymes, reactions will occur too slowly to sustain life. Okay, muscle contraction will occur very slowly without enzymes, or if there are few enzymes involved only. Right? Movement will also occur slowly. Maybe you will move like may move like an amoeba without enzymes, because he can't produce enough energy, quickly for muscle contraction. Speech will also be very slow, right? You want to say something also, it will come out very slowly. Like I want to eat nasi lemak. Everything will be slowed down. All right, if we live in a world where there are few or no enzymes. So we need our enzymes for efficient metabolism and production of energy, efficient production of energy for living processes. Learning standard number 3. Describe the naming of enzymes with the addition of ASE to their substrates. So enzyme nomenclature, meaning the system of naming enzymes, is based on the substrate or reaction that it catalyzes, right? So we're going to see how it's based on the substrate. So this method of naming enzymes was introduced by the International Union of Biochemistry and Molecular Biology, where the name of the enzyme is derived by adding the suffix ase to the name of the substrate that it catalyzes. For example, if your substrate is lactose, what is the name of the enzyme that catalyzes the breakdown of lactose? So add ase to the name. Take out ose and change it to ase. So the enzyme which catalyzes the hydrolysis of lactose, the substrate, is named lactase, right? So lactose plus water, and this breakdown is catalyzed by lactase, produces glucose plus galactose. Okay, so this reaction or equation shows the breakdown of lactose into 2 monosaccharides. Okay, from a disaccharide, 2 monosaccharides are produced. So remember in the last chapter, chapter 4, we've already studied the breakdown of disaccharides like lactose. And the memory aid was Lady Guga, just to help you recall, memory aid. La for lactose, and then Gu is for glucose, and Ga is for galactose, right? And the enzyme is lactase, right? So let's practice naming the enzymes for maltose. What is the enzyme for maltose that breaks down maltose? Another disaccharide. Yes, the enzyme is maltase, all right? So remove the OSE and replace it with ASE. What's the enzyme for sucrose? Sucrase. What is the enzyme for lipids? Lipase. Okay? In this case, you remove the IDS or ID and put ASE at the back. So if you ever see a substance with a name that ends with ASE, if you see the name of a substance, it has the ASE at the end, it's very likely that it is an enzyme in the body. Okay, an enzyme that speeds up the rate of biochemical reactions. Now, a few enzymes do not follow the naming system that we discussed just now. Examples are enzymes discovered before this naming system was introduced, such as trypsin, pepsin, rennin. Okay, you notice these enzymes, they have another system of naming where the name of the enzymes end in IN, right? Okay, so they are following another type of naming system, right? And these are the exceptions. Okay, let's look at some terms. What is the meaning of the word substrate? So a substrate is the substance needed for an enzymatic reaction. A substance needed for an enzymatic reaction, and it is the reactant of enzymatic reactions. So usually in an equation is on the left hand side. And then after the enzyme acts on the substrate and catalyzes a reaction, products will be formed. So a product is the substance formed at the end of the reaction. And we are going to look at the lock and key hypothesis afterwards. So this hypothesis, okay, uses the key to represent the substrate, and the lock, like the lock that we use to padlock our doors, our grill doors, for example. A lock represents the enzyme. Okay, key represents the substrate in the enzymatic reaction. Okay, so this lock and key hypothesis was proposed by a scientist long ago, and it explains the mechanism of enzyme action, how the enzyme acts in a reaction, and then why enzymes are highly specific, and how enzymes remain unchanged after a reaction and can be used again. It also explains that the active site of an enzyme, which is in the lock, it's the lock with a hole in there to fit in the key. So the active site is a sort of depression in the enzyme, a pocket or a cleft in the enzyme. It has a fixed shape which matches exactly the shape of a specific substrate, okay, which can be compared to a key. Okay, so substrate can be compared to a key, and enzyme can be compared to a lock, okay, that we use to lock our doors or cupboards or whatever. So let's have a look at this lock and key hypothesis in a short while. Now, before we talk about the lock and key hypothesis, let me ask you, in your mind, how do you picture an enzyme molecule to be like? What is the structure of an enzyme molecule? So we have to build a clear picture of how the enzyme should look like. So if the enzyme is a protein molecule, it's a complex protein made up of a few polypeptide chains that are twisted and folded together to form a three-dimensional structure or shape. So it's usually globular, like a cluster of grapes, where the grapes represent the atoms or the molecules in the enzyme. Okay, and then in the enzyme itself, there is a depression or a pocket or an active site. Okay, it's called an active site or a cleft with a specific configuration or shape, like this shape here. Okay, this is the active site with a specific shape that complements or matches the shape of a specific substrate molecule. Okay, so here we see two substrates, substrate A and B. Now, which one do you think has a shape that matches, sepadan dengan, matches or complements the shape of the active site? Here, is it substrate A or B? You can see, I'm sure you will say that it's substrate A, right? Not okay. So substrate A, now the substrates have their own shapes, and substrate A here has a shape that fits into the active site of this enzyme molecule. When it fits into the active site, then it forms an enzyme substrate complex, and then the reaction can be catalyzed, and then the substrate may break down into its products, okay, which will then leave the active site. So the binding of the substrate molecule to an active site of an enzyme is specific, like a lock and key combination, based on the shapes of the active site and the shape of the substrate molecule, right? So only one specific key, if you're talking about lock and key combination, only one specific key can fit into a specific lock based on shape. Sometimes when you want to open a lock, then you got so many keys to try. You have to try it one by one until one key fits in, and then when you turn it, it will open the lock, right? So because the shape of the key matches with that of the lock, okay, then the lock can be opened. So likewise, only one substrate can fit into the active site of an enzyme, and then catalysis will occur, and then maybe the breakdown of the substrate will occur, right? So it's all based on the shape of the substrate and the active site. They must be matching, right? The shape of the substrate must match that of the active site. So in this way, an enzyme is said to be specific in its action That is, usually each type of enzyme acts on only one substrate to catalyze a reaction. Okay, because only one type of substrate can fit into the active site of the enzyme based on its shape. Let us now discuss the lock and key hypothesis. So, learning standard number 6. Explain the mechanism of enzyme action using the lock and key hypothesis. What is this lock and key hypothesis? It is just a proposal or an explanation given by a scientist long ago about how enzymes function, how they act. So it's called the mechanism of enzyme action according to the lock and key hypothesis. Alright, so this part is quite important for SPM, for the structured question, essay question, even the objective questions. Now first we must mention that the enzyme is represented by a lock and the substrate is represented by a key. Okay, as you can see in this picture, the enzyme is the bigger molecule and it has a depression or active site. Just like a lock has a hole in it, alright, for the key to fit inside. Okay, so that's why the enzyme is represented by the lock, the bigger item, and then the substrate is the one that fits into the active site of the enzyme. So it's considered the key that fits into the lock. Okay, so remember that enzyme is represented by a lock and substrate is represented by a key. Remember Suki, substrate, key. Suki is a Japanese name for a girl. Okay, now let's continue. So how we're going to explain the mechanism of enzyme action? Okay, take it like a story. Firstly, a specific substrate approaches the enzyme. For example, when these molecules are moving freely in the system, they may randomly collide with one another. Okay, that's how the substrate may approach the enzyme. And if the substrate here, the blue one, the blue shape, has a shape that is complementary to the active site here, that is in the enzyme, on the enzyme, then the substrate can fit exactly into the active site. Okay, so the substrate combines with the enzyme at the active site if both the shape of the active site and the shape of the substrate are complementary or matching, right? So an enzyme substrate complex is formed. What is an enzyme substrate complex? It's just a combination of the enzyme molecule and the substrate molecule. All right, but it's very important to occur because when these two components fit together like this, the enzyme and the substrate, then a reaction can occur, can take place. The enzyme can catalyze the reaction, which could be the breakdown of the complex substrate molecule into two parts, which are called the products. Okay, so a reaction takes place and products are formed, and then the products will leave the active site after the reaction ends. Okay, and then what happens? The enzyme can be reused again because it has not changed in shape. It has not changed its shape or its structure at all. So it's still the same old enzyme and can be reused. So that's why enzymes are needed only in small quantities, because they can be reused again. So what does this lock and key hypothesis show us clearly? Firstly, it shows us that enzymes are specific in their action because the shape of the active site must match the shape of the specific substrate for a reaction to occur, for the enzyme substrate complex to be formed and a reaction to occur. So it shows that enzymes are specific in action, and it also shows us that the enzymes remain unchanged after a reaction. Okay, and of course overall it shows us the mechanism of enzyme action. Another thing that may show in the photograph or in the diagram is that the enzyme action is reversible, can go forward or backward actually, depending on the conditions. Okay, but the main thing that the lock and key hypothesis shows us is that enzymes are specific in their actions or in their reactions. So try and memorize these steps in the mechanism of enzyme action. Let us look at an animation of the mechanism of enzyme action using the lock and key hypothesis. So firstly, the substrate approaches the enzyme. Now normally the enzyme and substrate molecules are moving about freely and randomly in the system because of the energy that they have. All right, so substrate approaches enzyme, and next substrate combines with the active site to form the enzyme substrate complex here. Now, once the enzyme substrate complex is formed, a reaction takes place and products are formed which then leave the active site as the reaction ends. Then the enzyme is reused again to catalyze the breakdown or the reaction involving another substrate. Next, let's answer learning standard number 4, which is explain the characteristics or the properties of enzymes. So there are 8 characteristics here or properties that we have to discuss and remember. Okay, number 1, enzymes are biological catalysts that speed up biochemical reactions in a cell. Okay, this has already been discussed. Secondly, they act rapidly. So everything about enzymes is fast. The actions are fast, and their main aim is to make the biochemical reactions in a cell go faster to sustain life. Now, number 3, the action of enzymes are specific. This has also been discussed, right? And it's important for SPM. The questions, this type of question can be asked in SPM to explain why enzymes are specific. So, firstly, what do you mean by the word specific? It means that each type of enzyme can act on only one substrate to catalyze one reaction. Okay, and the reason is because each enzyme has an active site, as shown here. The blue color lines represent the active site of this enzyme with a specific shape, like an inverted V shape like this, that complements a specific substrate, matches a specific substrate, like substrate A. So when the shape of the active site complements the shape of the substrate molecule, then the substrate molecule can fit into the active site like this, and then catalysis can occur. Okay, the reaction can be catalyzed and can occur quickly. So only one substrate with a shape that complements the shape of the active site, that matches the shape of the active site, can fit into and combine with the active site, leading to the catalysis of the reaction. All right? And leading to the formation of products. So if the shape does not match, if the shape of the substrate does not match the shape of the active site, then there is no reaction because the substrate will not be able to fit into the active site. Okay, like this. Cannot fit in because the shape of the substrate molecule is not matching to the shape of the active site. So no reaction occurs in that case. Let's take a quick look at a common question in SPM. So the statement is enzymes are highly specific, and you're asked to explain the property of enzymes as stated above, or the characteristic of enzymes as stated above. Or you may be given a picture of the lock and key hypothesis to explain. Okay, so the lock and key hypothesis basically explains how enzymes are highly specific, right? So to start off, first of all, define what you mean by highly specific, or what it means by, discuss what it means by enzymes are highly specific. So enzymes are highly specific in that each type of enzyme can act on only one specific substrate to catalyze one specific reaction. And then you have to explain how this occurs. So this is because each enzyme has an active site with a specific configuration. For example, it's shown here, the yellow part is the active site with a specific shape or configuration that complements or matches a specific substrate molecule. Okay, because substrate molecules also have different shapes. So when the shape of the substrate fits into the active site, catalysis occurs. For example, like this, right? The shape of the substrate, the red shape here, fits into the active site of the enzyme here, right? So catalysis occurs. And the binding of the substrate molecule to the active site is specific, like the lock and key combination in the lock and key hypothesis, okay? Where the lock is represented by, the enzyme is represented by the lock and the substrate is represented by the key. So give an example. For example, only starch molecules and not other substrates like protein or lipid can fit into the active sites of the enzyme amylase for hydrolysis to occur and for maltose to form. Okay, because starch molecules are the ones that can fit into the active sites of the enzyme amylase. So the reaction occurs then, where the amylase will break down the starch molecules into maltose. Going on to the fourth characteristic of an enzyme. All right. So the structure of enzymes remain unchanged and enzymes are not destroyed after a reaction. Now, based on the lock and key hypothesis, here were the enzyme and this yellow structure is the substrate. So when the enzyme has catalyzed the breakdown of the substrate here, the products are formed and they will leave the enzyme molecule. And here we see that the enzyme molecule remains unchanged, all right, based on the lock and key hypothesis. So this enzyme can be reused again to catalyze another reaction, all right. So enzymes are only required in small quantities and are reusable, as can be seen from the lock and key hypothesis. They are not used up at the end of a reaction. So number 4, structure of enzymes remain unchanged. They are not destroyed after reaction. Number 5, they are needed in small quantities, reusable. Most of the reactions, number 6, most of the reactions catalyzed by enzymes are reversible. They can either go forward or backward. All right, now let's take an example of this reaction, enzymatic reaction where starch is hydrolyzed, right, to form maltose. So this reaction is catalyzed by amylase and it can occur in the mouth, right? So the reaction can go either way, according to these arrows. It can go forward or backward. Now, usually in our human body, it goes forward. It's based on conditions that are present. Okay, so this is one example of digestion, right, where the enzymatic reaction is actually reversible. But in the digestive system, normally the reaction goes forward. That means starch will be hydrolyzed to form maltose. Next, some enzymes need cofactors to work more efficiently. Okay, and these cofactors are actually helper molecules that bind to enzymes in order to help them, assist them in their enzymatic reactions, right? And examples of cofactors are vitamin B and magnesium ion. So according to the definition, a cofactor is actually a non-protein compound or metallic ion like magnesium, okay, that is required for an enzyme to act as a catalyst. So they are like helper molecules that assist the enzyme so that the enzymes can work more efficiently. So these are good guys, right? The good guys. Now the bad guys are called inhibitors. So enzyme activity is slowed down or stopped by enzyme inhibitors. Examples are heavy metals like lead or mercury. So if a person were to have a high level of lead or mercury in the body due to exposure to these substances, then his enzymatic activities can be slowed down. The enzymatic activities in the cell, all right, enzymatic reactions will be slowed down. So this can lead to some health problems. That's why lead and mercury are toxic to the body, okay, because they are inhibitors that either slow down or stop the enzymatic reactions in the body, okay, and they can cause health problems or, for example, mercury poisoning. So it's advisable to limit exposure to heavy metals for the sake of health. So here's a useful memory aid to remember the 8 characteristics of enzymes, all right? So the summary of all the characteristics of the enzymes are as follows. It is affected by inhibitors, it has cofactors, an enzyme remains unchanged after a reaction, it can speed up the rate of chemical reactions, it is specific in action, needed in small quantities, is reusable, after the reaction can be reused again, and the reaction of enzymes involving enzymes are usually reversible, and enzymes act rapidly. Okay, these are the summary of the 8 characteristics of an enzyme. So our bio key is ICU, and then 3S. So Sir cube, 3S and 3R. So it's like I see you, saya nampak anda. Sir. So maybe you think of a teacher that you call Sir. I see you, Sir Cube. or Sir Q. So that covers all the characteristics of the enzymes. Okay, I see you, Sir Q. All right. So it's up to you whether you want to remember this memory aid or not. You feel that you can remember the characteristics of enzymes easily, then you don't have to use it. Now, classification of enzymes. So enzymes can be divided into two types based on where they function. Okay, that is, and these are intracellular enzymes and extracellular enzymes. So intra means inside. That means these enzymes are produced in the cell and retained in the cell for the cell's own use. They never get out of the cell. Okay, they never go out of the cell. Whereas extracellular enzymes, the word extra means outside the cell. So these enzymes are synthesized in a cell, but they are secreted out of the cell, outside the cell, and they are used outside the cell for external use. Okay, that's the difference. So examples of intracellular enzymes that are retained in the cell for its own use is the hexokinase enzyme used in glycolysis during cellular respiration. Now in chapter 7, we'll study about the process of cellular respiration, and it involves one process called glycolysis, where glucose is split up into smaller molecules. So this reaction that occurs in the cell is catalyzed by the enzyme called hexokinase enzyme. Okay, and the second example of intracellular enzymes are photosynthetic enzymes found in the chloroplast of plant cells, which carry out photosynthesis. So photosynthetic enzymes catalyze the process of photosynthesis, which is made up of which consists of a series of reactions. So where are intracellular enzymes found? They are found in the mitochondria, in the organelles like mitochondria, chloroplast, okay, and also found in the cytoplasm and in the nucleus of the cell. Now for extracellular enzymes, examples are trypsin. So trypsin is produced by pancreatic cells or cells of the pancreas, but after that it is secreted outside the cell, okay, into the duodenum or small intestine to break down the polypeptides during protein digestion. So enzymes involved in the digestion of food in the alimentary canal, they are all extracellular enzymes. So other examples are pepsin, amylase, okay, and other enzymes that are secreted into the digestive tract to digest food. So where are these extracellular enzymes found? One example is that they are found in the digestive system, in the alimentary canal. The next learning standard is communicate about the involvement of specific organelles in the production of A, intracellular enzymes, and B, extracellular enzymes. Simply put, it is that we must be able to explain the involvement of the function of organelles in the production of extracellular enzymes and intracellular enzymes. So how are the extracellular enzymes produced? So the production of the extracellular enzymes involves some components of cells or organelles that are used in the sequence below. First, the ribosomes will synthesize or produce the proteins. Next, the proteins are transported by the rough endoplasmic reticulum, which then form the transport vesicles to transport the proteins to the Golgi apparatus. In the Golgi apparatus, the proteins are modified and then enclosed in secretory vesicles, which move to the plasma membrane and secrete the enzymes out of the cells. So we will refer to the next slide for the full explanation, but here I would like to give you a bio key so that you can remember the sequence of organelles that are used to form the extracellular enzymes. Okay, this is Rudolph reindeer transports gold to secret place. Okay, so you know Rudolph the red-nosed reindeer, in this case is transporting gold to a secret place, all right, using the special kind of sleigh. All right, so let's look at the organelles here. Now for the production of extracellular enzymes, all these organelles are used, but for intracellular enzymes, ribosomes, rough endoplasmic reticulum, transport vesicles, and Golgi apparatus will carry out their function, but it will not involve secretory vesicles. Okay, for intracellular enzymes that are used inside the cell, they do not need to be secreted out of the cell. Okay, so no secretory vesicles are used to transport them to the plasma membrane to be secreted. All right. Okay, let's look at the production of extracellular enzymes. Right, let's answer this important question. Explain the production of extracellular enzymes in a cell. It is an important question for SPM, and we must know the process, understand the process, and memorize the process. Okay, so the first step is the ribosomes. All right, what happens to the ribosomes? So these are the ribosomes are the white dots shown here. So I've enlarged a few of them. Okay, and they are found on the rough endoplasmic reticulum. So ribosomes synthesize or carry out protein synthesis. Okay, ribosomes synthesize proteins or carry out protein synthesis, and they use this code from the DNA of the cell, from the chromosomes of the cell. They use the genetic code or instructions to carry out protein synthesis. Okay, so after that, number 2, the proteins will enter the lumen of the rough endoplasmic reticulum, which is the green structure here. It looks like a series of roads, okay, a network of roads. And the proteins are transported through the rough endoplasmic reticulum. That's the job of the rough ER, okay, or rough endoplasmic reticulum, to transport the proteins. So the proteins are represented by the red dots here, the red circles. Next, number 3, the proteins reach the end of the rough endoplasmic reticulum. So they are packaged in transport vesicles, okay, which are like tiny bags that contain the proteins. And then these transport vesicles bud off and come from the rough ER. They are formed by the rough ER, or rough endoplasmic reticulum. So they will be formed, and then number 4, they will move with the proteins to the Golgi apparatus. Okay, the next stop is the Golgi apparatus, which is the orange structure here. And the transport vesicle will fuse with the Golgi apparatus. So the function of the transport vesicle is to transport the proteins to the Golgi apparatus to modify, which will modify the proteins. Okay, so the transport vesicle is like a lorry, it's like a vehicle that transports the proteins from one place, that means from the rough ER to the Golgi apparatus. So what happens in the Golgi apparatus? In the Golgi apparatus, the proteins are modified into enzymes. So how are proteins modified? As an example, proteins can be modified by adding carbohydrates to it. Okay. Anyway, proteins are modified to form enzymes. Okay, we do not need to know what substances are added to it. All right, or how is it modified? And then they are packaged in secretory vesicles. Right? So the Golgi apparatus will package these extracellular enzymes that are formed into secretory vesicles. And then secretory vesicles will transport these proteins to these enzymes to the plasma membrane. Okay, so secretory vesicles will move to the plasma membrane, fuse with the plasma membrane, and secrete the extracellular enzymes out of the cell. Okay, to fuse means to join. Join, somehow it will join with the plasma membrane. Okay, so these are the 6 steps in the production of extracellular enzymes. If it is intracellular enzymes, the intracellular enzymes will not be packaged into the secretory vesicles, okay, but they will be sent to the internal parts of the cell. Okay, so secretory vesicles are vesicles that will secrete the enzymes out of the cell. So they are for extracellular enzymes. They are used to package the extracellular enzymes. So we have to memorize the 6 steps here and know the organelles that are involved. So all the organelles are in blue, okay, typed in blue print. And then we have also got to know the verbs like transported, packaged, modified, secrete, and so forth. Okay, these are also keywords in our answers. So copy down the steps in the production of extracellular enzymes. And for the diagram, let's look at the next slide to know how to draw the diagram of the cell. We don't have to draw these three-dimensional shapes here. Okay, we'll draw it in 2D. So let's have a look at how to draw the structures involved in the production of extracellular enzymes. So here is a 2D diagram for you to draw, and it's good practice to draw a diagram of the cell with all the organelles that are involved, like the rough endoplasmic reticulum and the Golgi apparatus, because you can be asked in the exam to draw these structures in a cell. Okay, so draw the diagram, and for the process of production of extracellular enzymes, you can use the notes that was in the previous slide. Okay, the 6 steps in the production of extracellular enzymes. So actually, now I'll go through the production of extracellular enzymes again very fast as a summary here. So firstly, actually, it's the nucleus that is the first organelle to be involved in the production of extracellular enzymes, because the nucleus has chromosomes with a genetic code on its DNA for the synthesis of proteins. So this genetic code are specific instructions how to synthesize the proteins. Okay, and the ribosome must get this genetic code from the chromosomes in the nucleus. So what is the molecule that's involved to copy the code from the DNA and then to send it to the ribosomes? Can you still remember from chapter 4? There's a molecule that's involved. It will copy the code from the DNA on these chromosomes in the nucleus, and it will bring out the code for the synthesis of proteins. Yes, the molecule is the mRNA, the messenger RNA or ribonucleic acid. All right. So messenger RNA is a shorter, smaller molecule than the DNA. It copies the instructions to make one protein, and then it comes out and goes to the ribosomes. All right, moves out to the ribosomes. Ribosomes then get the genetic instructions or code for the synthesis of proteins, and then they will synthesize the proteins. Next, the proteins, as shown here by the red dots, will move into the lumen or the space of the rough endoplasmic reticulum. So rough ER or rough endoplasmic reticulum transports the proteins through the lumen, and then packages, the rough ER will package the proteins in transport vesicles. Transport vesicles then transport the protein from rough ER to the Golgi apparatus. Okay, then in the Golgi apparatus, the proteins are modified to form the extracellular enzymes. Okay, then the Golgi apparatus will package the extracellular enzymes into secretory vesicles, which transport the enzymes to the plasma membrane. Secretory vesicles fuse with the plasma membrane and then secrete the enzymes out of the cell. Okay, so take note that the transport vesicle transports protein from the rough ER to the Golgi apparatus, but the secretory vesicles transports the enzymes from the Golgi apparatus to the plasma membrane. Lastly, let's look at an animation of the process of production of enzymes. So this white dot here represents the ribosome, and the diamond-shaped structure here is the protein that is synthesized by the ribosome. So firstly, the ribosome synthesizes the protein, and then the protein moves into the rough endoplasmic reticulum, which transports the protein. The rough endoplasmic reticulum transports the protein and then encloses it in the transport vesicles, which bud off from the rough ER. Then transport vesicles transport the proteins to the Golgi apparatus, and the proteins are modified in the Golgi apparatus to form enzymes. Then the enzymes are packaged in secretory vesicles, and secretory vesicles transport the enzymes to the plasma membrane. Then secretory vesicles fuse with the plasma membrane and secrete the enzymes out of the cell. Okay, so that's the end of our lesson today. Thanks for watching. I hope that you have learned something from this video. Do share, like, and subscribe. And we will continue with the next topic soon.